it's very important to think of this as like a leaderboard, let's say, right? You have your algorithms, you have the metrics for each algorithm, and then you have multiple tasks, and you have to see what you pick for which task. And I think that's also something that it's critical to have.
Hi everyone, welcome to Let's Talk Research, the official InstaDeep podcast where we dive into cutting-edge AI research and the stories behind it. My name is Bora Güloğlu and I'm a research scientist here at InstaDeep.
And I'm Massimo Bortone. I'm also a research scientist at InstaDeep. Today's episode is a bit different than the usual ones we had so far. Instead of looking at a particular machine learning innovation, we will be talking about a real-world research challenge. In particular, we will be looking at how to design a protein that binds to a specific target, in this case, the Nipah virus.
Exactly. And just for some context, InstaDeep recently placed first and second in a challenge run by Adaptyv for protein design. And we have with us today for the discussion two people who are very highly involved in this project. We have our synthetic biology lead, Liviu Copoiu, and our immune structures lead, Antoine Delaunay. Welcome both.
Glad to be here.
Yeah, thanks for having us.
So guys, we do this every time on the podcast, you probably know. Before we start talking about the science and the challenge, let's hear a bit more about you and how you arrived at InstaDeep.
Sure. So my name is Antoine. I arrived at InstaDeep 4 years ago. I joined as an intern, and since then I've always worked on protein design and protein structure prediction. And my background is actually in applied maths and statistics. I graduated from Polytechnic and Imperial College.
Yeah. So for me, I joined also around 4.5 years ago, more or less. I joined for structural biology based projects. I started with kind of like looking at the basics and then how to build them up for applied projects. My background is in structural biology. I have a PhD in protein engineering, and it's so ancient that AI algorithms weren't really a thing for biology back then. I think ESM1B was published right when I was finishing my PhD. So I kind of like went along with the flow of information and innovation. And since then I'm kind of trying to apply whatever it's happening in the field for specific projects, same as my colleague Antoine.
As you kind of hinted a little bit, Liv, obviously there's been tons and tons of research in this area and lots of leaps in the past few years. And the big thing that we've seen is essentially that we are now seeing a level of maturity from these models, from these technologies, where we can actually apply them to protein design and protein engineering. Just for some context, would you mind explaining a little bit what you understand when I say protein design, protein engineering, and so on and so forth?
Yeah, so protein design and protein engineering are, like, for me, two related things, but are not like completely equal, right? But I think, so protein design is really you are trying to design a protein, and usually we say de novo protein design, so really try to design it from scratch, right? Whereas protein engineering, you can just say you want to engineer a specific property of your protein. And down the line, the ultimate goal is to drug, right? To make a drug for a given target that you're trying to bind to, or you can, sometimes you also already have like your protein and you just want to improve it. So you're going to say you want to make it more soluble, or, like make it bind more strongly to your target. Or
I think if there would be anything to add, I would say that, protein engineering is kind of like the old school, right? Of looking at something that you already have. And sometimes you try to improve it for binding. Sometimes you're trying to disrupt some binding, or sometimes you even try to make it more stable, right? While de novo, basically, design means de novo, which, at least for me, which basically means kind of like, oh, I have this target. Let me find something that is not found in nature that binds to it, right? And something to that something that you said is like, these models are maturing, but in our kind of like hands, they're still kind of like behaving as children, to be honest, because it's very easy to say, oh, this model behaves like this on this benchmark, which is synthetic and artificial, and then another algorithm comes and says like, oh no, I'm better because I've done this and this. And I think the only way to actually see where these models are is to kind of like on each task, right? Because now we have quite general models. Is okay, somebody comes up with a challenge, and then you just go blind and you say, okay, I have a month. Let me see how good I am.
Mm. And from an application point of view, is there like what's the benefit from doing de novo design compared to engineering a pre-existing protein?
Well, it's a bigger design space, right?
Mm.
So humans only solved a small fraction of what's possible because they are only interested in certain aspects, right? So if you think of the landscape, right, let's think in 2 dimensions, we barely scratch the surface, right? We only have like 250,000 structures solved. But if you look at how many sequences you have in UniProt or in other databases, it's hundreds of millions, right? So you don't have that much structural information to kind of like build upon it, right? Obviously, AlphaFold made a huge leap forward, and now we have the AlphaFold DB and other databases, the OpenFold one, and so on. So we get more and more information, but in reality, de novo just gives you more design space. That's pretty much.
But it also sounds like it's a much more computationally demanding problem, right? Than protein engineering.
You need to solve like both the structure and the sequence at the same time. You need to get everything right. Also, if you start from something that already works right and you try to engineer it, it has way less chances of having any sort of like toxicity issues down the line, right? Whereas if you design something from scratch, you need to make sure that, okay, your protein folds, your protein is going to target like what you want it to target, right? But then you need to make sure that it doesn't target anything else, that it doesn't bind to anything else. So with de novo design, you have like much more possibilities, but then you have also to think about a lot more problems at the same time. It's like an optimization problem with a lot of objectives at the same time, and some of them are a bit contradictory actually. So this is why it's so difficult.
Yeah, and I think also since we talk about applications, it also matters what type of application you have in mind, right? So are you thinking about creating something that will be expressed in a human, or like part of like of a vaccine or something? Because then the threshold is much higher on what you need to do. Are you thinking about tools for analytical research? Then the bar is much lower. So I think for de novo design, the field is like quite hyped, right? About like, oh, we can do all of these things with these algorithms, but it's still trying to find how can we actually make this into a reality that is useful beyond, nice papers, nice proof of concept. And I think that's the difference between de novo and so design and protein engineering or affinity maturation or property improvement, because we already see kind of like this work of like, oh, let's like the most classic one, let's say, would be like kind of like humanization, right? Where you have these framework regions that are classic and they are okay, and then you graft CDRs, right? Like that would be like a very old form of looking at protein engineering. So the de novo field and the design part is still not yet kind of like finding its place for applications.
So just to flesh that out a little bit more, I'd be quite keen to hear about how you approached the Nipah virus competition, and kind of what tools you used and what your general thinking for strategy was there.
Yeah, so we had like 2 main strategies, a bit like, I think it links back to what we were just saying, right? So one strategy was essentially lead optimization. So we had an existing binder and then we tried to optimize it further and further until we reach like a decent threshold. And then we also did de novo design, which is, as we just discussed, a much more difficult task. And so, in terms of tools, we used all the latest tools that are open source out there. So BoltzGen, RF diffusion, Protein PNN. And then it's also where I think like human expertise really helps to use these models, because we know from the benchmark that yes, they work in general, but then when you apply to a very specific target, then sometimes it's a bit more complicated, right? Because it can have this sort of like target dependent performance. So you really need to have also a human eye, and that sort of like checks and brings all these components together if you want to be successful. And I think we were quite successful, because I think we reached 0.37 nanomolar for the lead optimization. And for the de novo design, I don't remember how many we had.
So I think we had one binder and we reached like 200 nanomolar, I believe. So kind of like not at the level that we had for the lead optimization, but also very few sequences were tested. Usually you know like one test like hundreds, right, to kind of like get something, and we had I believe like 20 or 30 tested. So that's a difference. And if I can touch on something that Antoine said, so having a human in the loop is still necessary. There's no way you can do it. And I don't mean a human who presses a button or makes sure that you don't have errors, or that allocates enough resources. I mean an expert who can look at a sequence, at a structure, and identify like liabilities. So this is kind of like part of also, let's say, like our skill set. On top of that is we're using open source algorithms and ideas from the open source community, but we're also modifying everything in order to fit our purposes, our tasks. So it's very important to think of this as like a leaderboard, let's say, right? You have your algorithms, you have the metrics for each algorithm, and then you have multiple tasks, and you have to see what you pick for which task. And I think that's also something that it's critical to have, and not pigeonhole on a single method. So I think that's a bit of kind of like how we approach the problem. And Antoine talked about binding, which is kind of like the main thing that we are interested in, but there was also kind of like a secondary goal was to reach kind of like some sort of neutralization, right? So basically your molecule binds so strong to the target that the viral protein cannot like actually bind to its receptor and get inside our bodies. So obviously this is all done in vitro, but our two lead candidates, right, the ones that ranked first and second overall, they both achieved full neutralization. So kind of like that also speaks a bit about how we're thinking, not about just how do we get the best binding, but also what else is important downstream.
I mean, this echoes a little bit what we discussed in the last episode, which was on MLIP, so somewhat unrelated to the discussion. But again, there was this point that we kept coming back to, which is around kind of having a human expert somewhere in the process who's able to actually pick what tool to use for what task. And that really shines through in every application of ML. And in terms of validation, I'd like to kind of revisit this point of validation experimentally. So when we obviously look at in vitro metrics, we're often looking at affinity, which is kind of how tightly two molecules associate with each other. And that's great, but obviously in the context of biology, there's a lot more things that are happening. And Antoine, you also mentioned that kind of this is a big problem, especially during de novo design. So what value do you see in kind of having these big experimental benchmarks basically that you can
Yeah.
use.
So I think this is very important because so you have like existing benchmarks, like open source benchmarks, right? And most of the time they rely on very clean curated data sets, but at the same time they can be very biased because so they can be biased toward like specific types of proteins, or sometimes you have issues like the test set is too similar to the train set. It's not like completely properly split. So then the performance, when you assess the performance of the model, it's a little bit inflated compared to like a real world setting, right? And this is why we also say, oh, we still need the human in the loop as well, because then yes, sometimes it works on the paper, and then you apply it on your real test case, and then you realize it doesn't work. So then you need to think about, oh, how can we improve it, right? So I think these like public competitions are very good because they put everyone on an equal footstep. All the sequences that are submitted by all the teams are assessed with the same experimental method, which is also another problem, right? When you deal with public data, sometimes different methods, different experimentalists and so on. So here, same method, same conditions, same metric and so on. And then you assess it on a completely new target, right? So you make sure that it's not been seen previously and so on. So it's like really like a good hard test case. And then because then you have the ranking, then you can see, okay, like the team who used this model performed better than this model. So, then you can sort of like analyze the results. You can, and compare to what you've done. And then just, start this sort of like feedback loop, right? And I think that's like the key thing. Yeah.
So you're saying the community as a whole learns a lot from the outcome of the competition itself.
Yeah, and not only kind of like it shows about the models, but also about the strategy, right? Because for this competition, some people went with affinity maturation, and some people went with the novo design, and it looked right. From the results that we got that. The affinity maturation gives superior binding, right? Binding affinity. But, kind of like, why is that? Is it because the strategy for binding affinity is more mature, then it's more clear what you have to do? While for the novo, maybe you need to test more. Maybe you need to kind of like do multiple rounds. Like, what are what is the threshold, right, for diminishing returns one way or another? So I think it's kind of like good to see what different people are doing from a strategic point of view, to say, okay, maybe that's interesting. Maybe next time I can think about this because I didn't right before. When you read a paper which passed like 3 rounds of review and it's the work for like 2 years, you really have time to think about everything in a very controlled environment. When you get a competition in which you need to submit in like 4 weeks or 6 weeks, then you kind of need to be production ready, right? You need to have something in place and you need to run it, maybe do 1 or 2 iterations in silico and then send it. Right, so there is not enough time to kind of like, oh wait, let's try this, let's add 100 more constructs that we can test. So that's kind of like the beauty of these competitions, which are not cheap, right? Yeah. For the organizers, and that's why kind of like it's very important to support these efforts, even not by participating, but by kind of like sharing their efforts and kind of like trying to be part of the community, right? Like even like post about it, you go to a workshop or to a conference, try to engage and say like, oh, we participated here, and they are doing this recurrently. So I think it's just good to raise awareness about this stuff.
And I think there's a long history of this sort of competition leading to actually big breakthroughs in science also. I mean, AlphaFold came out of a similar kind of mechanism.
CASP.
CASP
is probably the best and biggest example of how a properly set up challenge can lead to data that is uniform, that is hard, and that leads to leaps in technology. And we need these types of efforts in order to push the field forward. So
yeah. So I've personally never taken part in a competition like this, but I'm curious about how sort of the human aspect is. So after the competition, how do the different teams talk to each other? What's the tone? Are people
Well,
hopefully well
so
behaved?
it's kind of like we didn't have a forum to discuss, right?
Okay.
Let's say, but like with other teams,
Yeah.
right? And we also didn't know who participated, right?
Oh, right.
So beforehand, right? So it was kind of like, like internally, we kind of like had weekly meetings, right? Like discussing strategy and discussing what we can do. We were also like a very small team. So like, I think in total, like maybe 6 people were involved, but they were different kind of like levels of engagement, right? To this. So we had Miles and Nikita, who were kind of like the leads for the IC work. So Miles was leading the affinity maturation, and Nikita was leading the novo part, while more senior members were kind of like trying to find out what would break, validating, looking through kind of like what strategies are viable and so on. So I think we have, by working on so many applied projects, I think we have a very good understanding of how the team dynamics need to happen when you have to deliver quick, right? And I think this is also an advantage that we have at InstaDeep. So it's kind of like this. I wanted to touch on one point that Antoine actually correctly made, is that it puts everybody on same footing, and it generates data that is uniform, right? It's on the same instrument at the same time, because what the field and a lot of papers are kind of like doing is they try to aggregate data. And even if you aggregate data from the same type of experiment, so let's say, you have an SPR that does affinity calculations, right? And measurements. If you do it if another group does exactly the same thing, they can get a lot different results, because maybe the construct was a bit different. Maybe they didn't immobilize enough material, or maybe they even used another chip, right? Like it's there's a lot of variation in biological experiments, and this is without including cells, right? The moment you start to look at experiments that have like something to do with cells, then it's even more blurry and more noisy. And I think this is the difference between, LLMs like, ChatGPT, Llama, and, what Anthropic is doing with Claude. It's kind of like they have the advantage of having the full breadth of everything written in the history of the world, right? Like everything that they could get their hands on, they got their hands on, right? And you have such a huge breadth of languages, types of writing, that it's kind of like you don't have to care that much, right? Like, you can do that, and then with smart tricks and tips, you get very good reasoning, very good question answering. But with biology, the data is so heterogeneous and so small compared to like what's possible, that it's kind of like very hard to believe that, that's a general model that can do so many things.
Yeah.
We're just not there yet.
And obviously one way to verify kind of how applicable one strategy is to different problems is just to apply it to different problems, right? And to test out different settings, different scenarios. Have you found in the team basically having also just set up this culture of fast iteration, and also just the infrastructure of being able to apply the same tools to different problem settings? Have you found this to be the case? To what extent do you kind of need to rejig a strategy when the problem changes, for example, from a general protein binder to, let's say, TCI engineering?
Yeah, so I think that's one of our strengths as well. So we have what we call our model garden, where essentially we combine all the models in one place, and then we can essentially chain them together. And so then when we have a new problem, and also based on the feedback we had for previous tasks, then before launching them, we think like, okay, what would be like the best model we want to try, and so on, what's our compute budget? And based on that, we make decisions. And then because we have set up all this infrastructure, and we have everything in place, then we can run things very smoothly and easily. And I think that's one of our key advantages, because, as Liviu said, like we don't have that much time for these competitions. And obviously we have like other projects going on at the same time. So if you want to participate and be able to, make decent submissions, then, you need to have these things in place so you can spend some time, but at the same time, just do your day-to-day work as well.
Mm-hmm. So guys, it sounds like you've been learning a lot throughout this competition. I'm curious to hear if you've applied any of these strategies and learnings in other cases and contexts.
Yeah, so I think if we're talking the novo, we've been working on nanoparticle design. So nanoparticle, the ones that we worked on are basically, think about protein kind of like cages that are symmetrical, and that they're made of like these tiny little pieces that just form together the cage. And the field has proven that nanoparticles are viable for vaccine design.
So these are used to deliver
so like a
drug to the
so
to the cell.
not a drug, but like an antigen.
Okay,
right? So like not a small molecule, but an antigen, right? And basically we wanted to tackle this problem, and a team at InstaDeep, which was kind of like led by Yongguan, Cheng, and Adam, they kind of showed that you can use our model garden to kind of like apply to a completely different problem the same algorithms, right? And they got experimental validation, and they showed kind of like, different ways you can make these particles with different kind of like components. And I think this was our first de novo design kind of like success with validation. And then we start building on that, right?
We also work on T-cell receptor affinity maturation. So essentially the idea is that we are trying to engineer the receptor of white blood cells. Mm-hmm. And these are going to target a protein complex called the peptide MHC complex, which essentially is complex that you can find at the surface of cancer cells, right? And so the issue is that you have these white blood cells that have this weak binding affinity because they're not supposed to bind to self cells, right? So you need to, if you want to make it work for cancer cells, then you want to engineer them so that they bind more strongly. And so what we're doing here is we start from something that we know works, then we can model the structure, and then we can find like the good mutations that are going to increase the binding affinity, while at the same time keeping the specificity. So meaning we don't want the T-cell receptor to become reactive to something else, right? And so we had like very good results. We've done that like several times. And we actually reached for 2 campaigns, the preclinical stage. And we had, deep in vivo tumor control on animal models. So that was like very nice results.
Yeah, and if I can say one thing, so we're discussing these 2 projects, but we're not doing this alone, right? So these are with our colleagues from BioNTech, and we've been kind of like blessed to have access to world-class experimental validation that is relevant for downstream applications. So whether we work with BioNTech US or BioNTech Germany, we're always kind of like in touch with wet lab scientists, with people who are in charge of like creating the next therapeutics. So just being in this ecosystem and not just, like on our own island doing AI, right? It helps tremendously because you learn what's important. For somebody who develops a drug, for somebody who's trying to innovate in this space of molecules, of biologics. So we would not be where we are without our colleagues at BioNTech. So I think that's a very, very important aspect of our work.
Yeah, and I think that's also a tremendous advantage, because then as you sort of like start collaborating and so on, then you kind of get this nice feedback loop where, we send the sequences, they validate them, we come back with the results, we can learn from that, and then we iterate like, step by step. And I think the more we iterate, then the better this like loop is, right? And the better the models are as well. So this is really nice.
Super interesting. And kind of, I'd like to get your thoughts a little bit on where you see the field moving, what sort of exciting kind of challenges there are ahead, but also opportunities, and kind of, what's been nice to see happen, basically.
Yeah, I think on my side, I'm quite excited for all these companies that are going to generate a lot of data at high throughput. We are seeing more and more of them. And obviously, this is going to fuel our AI models. And this is what's going to make our models much better, right? We've also seen like interesting initiatives, right? Like in the UK, there's the OpenBind Consortium, right? Public-private partnership. And they aim to generate hundreds of thousands of data points, right? And then to open source them to the whole community. And I think this is really exciting. This is gonna, represent, I think, like 10x fold, or I don't want to give an exact number, but compared to the available data, right? That's just gonna be huge. And I think this is really quite forward-looking, yeah.
Maybe to bring it back and relate it more to the work that researchers at InstaDeep are doing and the previous episodes that we had on the podcast. Where do you see the development in terms of machine learning, model innovation, and how, new models that you are curious about integrating into your strategies and work?
Yeah, so basically we've already started kind of like using some of the methods and algorithms that you guys discussed here in the past. So Bayesian flow networks, we work together with research on the Nature Communications paper, and then we work together on some validation, right? Experimental validation of the models. And now it is part of kind of like our model garden for certain tasks. So we're working towards doing this kind of like integration. And I think what's exciting for me is what is missing out there, right? In the literature, in the field, right? And for me, what's missing is adding some sort of physics-based information that is either a simulation or some descriptors to protein interaction prediction, and why stop there, right? Like protein RNA or protein DNA going forward. Yeah.
Brilliant. Thank you so much both for coming on the show today and for the fascinating discussion.
Thanks a lot for having us here today. That was great.
And if you'd be interested in playing around with or trying out any of InstaDeep's protein design tools, we'll link those in the description as well.
Thank you for watching Let's Talk Research Season 1. Stay tuned for future discussions from the brilliant minds behind our frontier AI discoveries and cutting-edge research. Goodbye for now.